this video clip, I'm going to showcase clinically used drugs that target DNA. Majority of drugs target proteins. However, there are many clinically used drugs that act on DNA. Doxorubicin is a highly effective anti-cancer drug. Doxorubicin is also called adriamycin. It is a naturally occurring antibiotic isolated from a family of bacteria called Streptomyces. Doxorubicin has a tetracyclic structure where 3 of the links are planar. This planar structure slips into double helical DNA between base pairs. Such interaction is called intercalation. The positively charged amino group also interact with the negatively charged phosphate of DNA. What you are looking at here is 2 doxorubicin molecules intercalating between base pairs of DNA. You can see their amino groups in blue right here and here. This interaction inhibits the action of the enzyme called topoisomerase II, which relaxes DNA structures for replication. The topoisomerase II is most active during cell growth. That's why doxorubicin is most effective against rapidly growing cells, including cancer cells. Some of the drugs react with the DNA bases. There are several nucleophilic groups in the structure of DNA bases, especially the N7 position of guanine. The N7 of guanine can be reacted with electrophilic reagents to form an alkylated guanine, shown here. Such a reaction is dangerous. It may lead to mutation of DNA. The guanine base is usually in a keto form. And this keto form permits hydrogen bonding with the cytosine base. However, when the N7 of guanine is alkylated, the guanine base tends to be an enol form, which prefers thymine as a partner. This mispairing leads to the mutation of DNA and may cause cancer. But DNA alkylating agents can also react with proteins and may be toxic. But nevertheless, some of the DNA alkylating drugs are useful in treating cancer. One example is cisplatin, one of the most commonly used drugs. In the list of 100 best-selling drugs, you can see a more optimized version of cisplatin, oxaliplatin or exosatin. The drug number is 34. Do you want to see its constellation? One ethics student wrote this. I like this because it is well thought. In Latin, cancer means crab. The finger-like projections of blood vessels from cancer look similar to the shape of a crab. Now, these 2 crabs can be removed by a platinum bomb here. Let me go back to the structure of cisplatin. Cisplatin has a platinum atom linked to 2 chloro substituents and 2 ammonia molecules ligated to a platinum atom. This is a stable molecule, but when administered into cells, the 1 or 2 of the chloro substituents are displaced by water molecules. the resulting positively charged molecule is reactive. The water ligand can easily be displaced with nucleophilic groups. In DNA, the N7 position of guanine is preferred. Typically, if another guanine base exists nearby, the cisplatin cross-links two adjacent guanines. The cross-linking reactions prevents cell division and promotes cell death. Another drug I would like to show you is bleomycin. Bleomycin is a natural product isolated from Streptomyces in Japan. This clinically used drug has a very interesting mode of action. This part of the molecule intercalates with DNA. The nitrogen atoms of the amines, pyrimidines, and indole ring here chelate a ferrous ion. Then here is the structure of an ion bleomycin complex right here. This structure is analogous to that of heme in your blood to transport oxygen molecules. In fact, the ion bleomycin complex interacts with oxygen and is oxidized to a ferric ion. The reaction generates superoxide or hydroxyl radicals which abstract the hydrogen atoms from DNA backbone. Here's a superoxide anion radical. The hydroxyl radical is like this. These are very, very reactive. The hydrogen abstraction from DNA backbone leads to the DNA strand breaks, which inhibit gene expression and replication. The nature is a great designer of molecules. In the next video clip, I will tell you about the best-selling cookies in the history of humankind.